Welcome, everyone, to this webinar on New Frontiers in Computational Protein Design and Structural Prediction, which is brought to you by the Protein Society. My name is Liz Meyring. I'm the president of the Protein Society, and I'm delighted to be introducing our webinar today, which showcases amazing advances in the field of the most recent Nobel Prize in Chemistry for protein structure prediction and design. Before turning this over to the chair for this webinar, Professor Alena Khmelinskia, I have just a few announcements. So I want to invite you to join us still, you can, for our annual symposium, which is next month in San Francisco. We have a stellar program, which you can see at our website, and you can still register and learn about the latest in protein science at this meeting. I also would like to highlight the date for next year's symposium, which will be celebrating the 40th anniversary of the foundation of the Society. And this meeting will be in Boston, July 19th to 22nd in 2026. And I want to highlight as well the journal for our society, Protein Science. We have an outstanding editorial team led by John Correia and the senior editors shown here, as well as an editorial board that covers a very wide range of first-rate protein science. So I encourage you all to publish your research here as well. And I want to highlight that the journal also provides Best Paper Awards. This is actually, all papers are considered for this automatically, and a junior author is designated as an award winner and is invited to give a talk at the annual symposium. And as well, there are other benefits that go with winning that award. So that brings me to our main event today. I want to warmly thank Alena and our speakers for sharing your exciting research in this amazing frontier field of research. I also want to thank our staff at the Protein Society, Raluca and Shannon, for their help behind the scenes. And I'll give just a brief introduction to Alena before turning the proceedings over to her. So she obtained her PhD in physics with supervisor Petro Schwille at the Max Planck Institute for Biochemistry in Martinsried, Germany, and then moved to the University of Washington, working with Neil King in the larger empire of David Baker, who is the 2024 Nobel laureate in chemistry for protein design. She then took up the Argelander Junior Professorship at the University of Bonn, and in 2023, her lab moved to the current location, the chemistry department at the Ludwig Maximilians Universität in Munich, Germany, where she holds a W2 professorship in biophysical chemistry. Her group focuses on dissecting the physical principles of protein self-assembly. They combine computational de novo protein design with protein production, in vitro models of model substrates, and biophysical methods to systematically investigate the interplay between different types of interactions in the assembly process of protein-based materials. So before we, Alena continues with this, some logistical notes. The way that we're going to do this webinar is we'll have these 4 presentations. Each speaker will present for 20 to 25 minutes, which will include a brief question period. After the talks, we may have some further questions in a discussion format. So I encourage you all now to take a moment to find your Q&A tab, and that you can type any questions you have there at any time. Only the organizers and the speakers will see them, so that's us here. The whole audience does not see them. And questions will be read out and answered in real time. And some answers may also be typed, and then they'll be visible. The answers will be visible in the chat. And after that, the webinar is also going to be posted on YouTube, so for your reference. So I'd like to thank all of you in the audience for joining us today. Your participation is a key part of the webinar as well, and it really enriches the discussion and the science. So we really welcome you to share your thoughts as well. And with that, I will turn it over to Alena.
Thank you, Liz, and thank you everybody for being online. It's a true pleasure to being able of organizing this webinar and for highlighting the research that is so close to all our hearts. And yeah, I just want to start with a few words of introduction to the topic, and with that, really, what I wanted to remind all of us is that in the few past few years, we really witnessed a technological shift with artificial intelligence flooding all domains of our life, from the most mundane tasks to a much broader impact in science. And this has really been recognized by last year's Nobel Prizes, not only for chemistry, as Liz mentioned, but also the Nobel Prize for physics. But this Nobel Prize for Chemistry to the Miss Hassabis, John Jumper, and David Baker was really particularly close to the heart of this community, the community of protein scientists, as it really highlighted the broader societal impact that it can have and on the community. And in specific, how structural prediction and protein design combined can be used to build new tailor-made proteins that will be useful for a wide range of applications from medicine and biotechnology. But as David himself pointed out in his Nobel lecture, this has been just the beginning and there is much more to come. And so in this webinar, as the title says, we're going to try to explore some of the new frontiers in protein design and structural prediction. And for this, I invited three great representatives, I think, from our community. So first of all, we have Minkyung Baek joining us, and she's an assistant professor at the Department of Biological Sciences at the Seoul National University. Her research focuses on developing artificial intelligence methods for predicting the structure and interaction of biomolecules from proteins to nucleic acids. And you may recognize her name as one of the main developers of Rosetta Fold, and she has also contributed to advancing AI-driven approaches for protein folding, design, and drug discovery. These days, her lab integrates structural biology, computational modeling, and deep learning to understand life at the molecular level. Then we have also Bruno Correia joining us. Bruno studied chemistry at the University of Coimbra in Portugal, and then did his PhD in Seattle with David Baker, and later his postdoc at the Scripps Research Institute with Ben Kravitz. And for the last 10 years, he has his research group at the École Polytechnique Fédérale de Lausanne, so EPFL, where he has been working on novel methodologies for protein design. And finally, I have the pleasure of having also Hannah Wayment-Steele with us. And she started this year as an assistant professor at the University of Wisconsin-Madison in biochemistry. And her lab combines computation and experiments to better understand and design biomolecular dynamics. So Hannah completed her PhD in chemistry at the Stanford University, where she worked with Vijay Pandey and Rihu Das. And more recently, she was a Jane Coffin Childs postdoctoral fellow with Dorothy Kern at Brandeis University and a visiting researcher at Google Brain with Lucy Colville. And so with this, just as a reminder, please participate actively. It really helps to have your input on the Q&A, so feel free to use the chat as much as you'd like. And to get us started, I would introduce first the research that we're doing in my lab. So let me try to share the screen. All right. Right. And so what we work in my lab is a lot to do with protein self-assembly. And before starting, I really would like to highlight the wonderful team that has been working with me. And in particular today, I will focus on the work of Ho-Young Chim, who is sitting here in the middle of the picture. And it has been a true pleasure to being able of working with these bright minds behind the scenes. Usually I like to start this presentation by highlighting the diversity of protein assemblies that exist in nature, just as a powerful reminder of how putting together smaller building blocks, we can generate functions that otherwise would not be possible by individual proteins. In this audience, of course, I don't have to convince anyone about the beauty of proteins, but I still really enjoy from time to time coming back to this picture and reflecting about the great diversity and functionality that nature had come up with these special machines that proteins are. Now, in my lab, we take the approach of designing self-assembly, and in particular, we focus on this type of capsid-like structures that somewhat resemble viruses. And these days, we are capable of designing them with some broad diversity, from 10 nanometers on the left-hand side of your screen to 200 nanometers on the right-hand side, covering a range of architectures from tetrahedrons over here on the left to octahedrons in the center and icosahedrons more on the right-hand side. And the reason that we are really interested in this type of material is that we have three different surfaces that make these particles really versatile as a material. And so on one hand, we can decorate the exterior of these nanoparticles with molecules to interact with the environment or with any cell that you are interested in. And here I show you an example from the King Lab where I did my postdoc. And this is a de novo design nanoparticle that is displaying the hemagglutinin of the flu virus, and is actually currently in clinical trials as a candidate vaccine against the flu. Besides the exterior, we can also take the opposite surface, so go towards the interior, and their possible inspiration are viruses that really take advantage of these interior cavities of nanoparticles to disseminate their genetic material. And so similarly, one can envision how one can use these microcompartments to protect cargoes from the surrounding environment, or even control reactions in a confined space. And finally, and probably one of the most interesting interfaces, it's really this inter-subunit interface that defines the properties of the particles from their geometry, the pore size, the volume, the radius, right? To really the functionality of the nanoparticle, as we can actually build in also properties within this interfaces or even within the subunits themselves to include functionality in the nanoparticle. And this is an example from the Baek lab, at the time from Robbie Devine, which is a nanoparticle that has two different components. And this blue component over here is the Fc domain of antibodies, so the constant part of the antibody. And now you can envision how we can swap this Fc domain by any antibody of interest to target particular receptors on a cell surface, for example. All right. Now, the way that we have been exploring this intersubunit surface is twofold in my lab. And the broader goal is really to try to expand the repertoire of particles that is available with two main aspects in mind. On one hand, the structural aspect, and there we have been looking in new type of architectures or using different building blocks like non-canonical amino acids, or even going towards the design of Lego pieces and introducing flexibility to expand the broadness of architectures that we can generate. The other aspect is more on the functional side, and there again, it's the idea of encoding in these protein-protein interfaces more than just structure, but including the functionality by, for example, including enzymes as building blocks, or going towards the design of metastable interfaces. Now, the story that I bring to you today is a combination of these two aspects, the structural aspect and the functional aspect, to how we went around to designing chemically induced protein assemblies. And chemically induced protein assemblies are not really new in the field. This has been explored previously. Some of the main examples are really towards the use of metal coordination, which can be rationally designed, or the introduction of pH sensitivity, where we've also done some contributions. But here, what I would like to explore with you is the possibility of designing chemically induced oligomers. And the way that we started by exploring this approach is, would it be possible to design small molecule dependent oligomers by taking advantage of existing or designed chemically induced dimers? And the way that we classically approach this problem is by taking existing building blocks and docking them into a conformation of interest. So in this case, a symmetric oligomer of cyclic symmetry type 4. So we have 4 repetitions of the 2 building blocks that interact through the small molecule. And this new interface between the building blocks we then computationally design to stabilize the architecture of choice. Now, Ho-Young Chim, when he came to my lab and I proposed to him this project, his immediate gut feeling was like, well, Alena, but this is not really gonna work, because typically we have architectures of building blocks that will not be just simply compatible with any type of new oligomer that we want to build. So the structure of the building blocks themselves will then influence which type of architecture can we actually build out of it. And so instead of taking this approach, Ho-Young proposed that, well, what we could do is take the interface of these chemically induced dimers, and instead of building a new interface, actually building a chimeric protein that on each side stabilizes one side of the interface, but the in-between area is what we have to design to then stabilize the angle between them to design the architecture that we're interested in. And he's really convinced that this works. And I hope that with the data that I present you, it also convinces you that this is a possibility. And so the way that we approached this was by taking advantage of one of perhaps most used models these days for backbone design. And this is RF diffusion. So it's a diffusion model. And diffusion models originally were trained to denoise images, so improve the quality of images. And the idea is that by training a neural network to denoise pictures to recover a completely clean picture, if we take it to the extreme of starting from complete noise, then the neural network would be generative in the sense that it would create images that recapitulate the properties of the images that it was trained on. So in that sense, if you trained it on pictures of cats, then it would also generate you pictures of cats. And indeed, this was what the Baker lab then did for training RF diffusion. They took the data that we have in the protein data bank, so in the PDB, and by training this neural network, created a generative version of it, where from Gaussian noise, it's possible to generate completely new protein structures without constraints or with constraints implemented. Now, in our case, what we are looking to do is something that also exists in the imaging world. So it's this idea of outpainting, where you can ask the neural network to complement the space around the image that you have. So basically to complete the image. And this is indeed what we want. By having the 2 sides of the motif, we want to be able of filling in the gap in between those 2 sides of the protein interface that we are interested in. And so the way that Ho-Young then decided to implement this was taking non-chemically induced dimer, extract the interface with the small molecule in it, so the 2 sides of the protein that interact with the small molecule, then symmetrize them and randomize the degrees of freedom that are necessary, so the 3 rotations and the translation in between the domains, and allow for RF diffusion to then fill in the space in between. Now, what we also did to improve the quality of the backbones, to avoid having 2 two domain proteins where the angle in between the two domains would not be so easily controlled, and rather move towards single domain proteins where everything is well packed and then the angles are concretely defined. We introduce this dragging force along the trajectory where we start with a motif perhaps in a suboptimal position, but then with the development of the diffusion trajectory, we are capable of moving the motif closer to where it would allow for this perfect packing of the motifs against the core of the protein. And so to benchmark this method, Wayment-Steele took three different types of motifs. So protein-protein interactions that have heterodimeric interactions that have been designed in the Baker lab, some metal-mediated interactions from the Akif Tajan's lab, and some small molecule-induced interfaces designed in Bruno Correia's lab. And so he chose a set of different assembly sizes. And to give you a bit of an idea, we started by covering trimers, tetramers, and pentamers, and ordered a diversity of designs in the order of 100 designs covering all these different types of architectures and these different types of interactions. In the metal-mediated interactions, we are talking about copper-induced interactions. And then in the small molecule case, one of the interfaces is dependent on venetoclax, anti-cancer drug, and in the other case is a progesterone binding motif. All right, and does this actually work? Indeed, our preliminary data really shows that this seems to work quite well. From all the designs that we ordered, the vast majority actually expressed very well and was purified at high yields, more so with the exception of the small molecule motifs, and we can discuss a little bit later why exactly is that. But just as a hint, mostly those motifs are much more complicated in terms of secondary structure, and in particular has a lot of loopy regions that are much more challenging to scaffold. But here I'll show you some data for just some example data for metal-dependent and protein-protein interactions. So we have a trimer from a metal-dependent interaction that in the absence of the metal should be a monomer. And then we have a simple protein-protein interaction trimer, tetramer, and pentamer. And indeed, if we look at our analytical side exclusion chromatography, we see that the elution profiles follow the expected behavior depending on the size of the oligomer that we have. Now, of course, the question is, are these also functional? So in the case of the metal-dependent interaction or even the small molecule-dependent interaction, and I'll show you the example for the small molecule case, where we have now mouse, so multi-angle light scattering data showing that the monomer without the small molecule, we indeed have a monomer in our, the protein does not self-assemble, but on the addition of venetoclax in this case, the trimer is formed, and it seems to be really responsive to the presence of the small molecule. And so the ongoing work here is really to continue our structural and functional validation. We have just off the press, just learned today that we have beautiful crystals for some of the samples, and we should soon be able to really look in more detail if the interfaces that we were scaffolding are perfectly aligned with our design models. So where do we actually want to go with this beyond just simply being able of designing these beautiful architectures? So our thought is really, well, if we are capable of designing this type of small molecule mediated or even metal mediated oligomers, then we can go one step further and start thinking about multiplexed sensing or even responsive nanoparticles that are assembled in one type of environment, disassembled in a completely different type of environment. So the idea here would be to somehow start breaking symmetry within the assembly so that we can have not just one single type of motif and one single type of responsiveness, but indeed being able of multiplexing several of these motifs and these types of sensitivity. And so one way that we started by approaching this is still assuming that we have only one motif of each species in our assembly. And there is two ways that you could think of doing this, simultaneous scaffolding. And one way would be to do this completely asymmetrically. So all the building blocks are different, and the interfaces are all different. But Hoyang wanted to go one step beyond that and actually introduce some sort of pseudo-symmetry at the backbone level so that it would be easier to then incorporate this type of assembly, this type of responsive assembly into larger architectures. And so the way that he does this is by, at the first steps of the diffusion trajectories, to enforce a resymmetrization at the backbone level only. So he takes one of the backbones that are diffused and he resymmetrizes at every single step. And then at the last steps, or the last n steps, he then allows for RF diffusion to do the diffusion completely asymmetrically. But now, because we are further along in our trajectory, these building blocks are simply readjusting to the individual motifs and less so changing completely their architecture. So in that way, we can achieve backbones that are very similar to one another. And by eye, one would almost call them symmetric at first glance. And the way that as a proof of concept, we are validating this on just simple protein-protein interactions. Again, these are from heterodimers from the Baker lab. And these are just some examples of of the type of backbones that he can generate. So in green is highlighted an example of a completely asymmetric diffusion pipeline where no constraints of symmetrization are introduced. And the other three are examples of where we are enforcing this type of symmetry. So something that was curious about this approach and sort of counterintuitive is that we actually could achieve much better designs by forcing the symmetrization at early steps. And here I'm showing you the data for 3 sets, 2 different time steps of enforced symmetry and the completely asymmetric case in green. And these are the results of predicting sequences of structural AlphaFold structural prediction of multiple sequences for multiple backbones. And what I'm presenting is the RMST values. So how well does the prediction align with the original backbone and the confidence of that prediction? And it was kind of surprising to see that AlphaFold 2 metrics for the completely asymmetric case seem to always be poorer than the ones for symmetric case on an overall glance, and made us wonder if there would be an inherent bias towards symmetry in AlphaFold 2, which we've also have seen in other contexts in the lab. Okay. So the other approach that we can take this is actually thinking about symmetrizing and co-generating multiple backbones simultaneously. So typically, a lot of the work in RF diffusion is still done on a single chain type of interactions, or perhaps 2 chains coming together, designing a binder against a particular target, but less so of co-diffusing 2 different backbones without having a symmetric constraint introduced. And so here we wanted to generate symmetric assemblies that would be of the type AB, 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 so that we would have several backbones in one single architecture. And this is what Harris Ip did during his bachelor thesis. So he introduced the necessary chain breaks within RF diffusion and fine-tunes the guiding potentials during the RF diffusion trajectory to really being able of having this spacing of interactions and controlling that the vast majority of the design backbones would indeed be of the correct conformation. And this is actually possible to do not just with two components, but with any type of symmetry. And here are examples from C3 up to C5 symmetry, and from 2 to 5 components. And we are now actually expanding this to 3 dimensions and beyond. Yeah. And with all of this, really where we want to go is this idea of off-the-shelf programmability. So the way that I usually talk about this is that I would like to have this idea of a vending machine for protein assemblies, where the user just simply has to input what are the properties that it should have, what it should sense, where it should disassemble, with which partners it should interact, and on the other side, something comes out. And as an example of that, I just want to give a shout out to one particular project that is done by undergrads in my lab. So in this case, Anna Baek and Elisabetta Maltseva. And this is what they call the duck and rabbit project, where the idea is that we want to design protein Lego pieces. So the same way that we look at this painting and the same set of lines can give us the impression of 2 different interpretations. So if we look that way, it's a rabbit, this way would be a duck. In a similar way, we can use the same structural motifs to then design either, in this case, a tetrahedral assembly, or on the other side, a dihedral assembly. So they have been hacking into RF diffusion and protein MPNN to really introduce 2 state design pipelines to being able of simultaneously optimizing our design to 2 goals at the same time. And so if everything is successful, then we are going to move forward to actually then additionally introducing conformational switches to being able of controlling towards which state are we actually biasing the assembly. And with this, I am at the very end. And I want to thank once again my team, the people that did the work, tried to already talk through the talk, and of course our collaborators, both in Bruno Correia's lab and Clarity Schroeder's lab, our funding sources, and once again, thank you all for joining us in this webinar.
Yay! I don't know what I'm supposed to do. I'm gonna clap. It feels strange to not clap. It's really fascinating, Alena. I wanted to maybe just start. I have a general question about your view on assembly in general, and so because one can sort of imagine constructing a big multi-component thing from distinct subunits, or from things being sort of on a single chain and co-translationally folding. And I'm curious like those both have pros and cons, and I'm curious your thoughts on that question in design.
Yeah. So our approach has always been to rather like repeat individual units, because then you can more easily actually get the folding right and making sure that your building block is what it's supposed to be. And then we design then extra interaction to build the larger scale assembly. But yeah, you can also indeed imagine that you would want to build a large protein to do things that are built from smaller pieces. And I think things like DHR, so the de novo helical repeats, are one example of how that may go, right? Where you just have this one repeat that repeats over and over again. But there has been some other cool work out there. So for example, from Roman Herala, they did the protein origami type structures with helical hairpins that then can also generate larger type of architectures. Or more recently, I think a very cool example comes from Hendrik Dietz' lab with this relaxed sequence optimization method, where they really went towards this like 1,000 amino acid large proteins. But they really did see indeed that like getting then the connections between the domains completely right becomes a much larger challenge, and you can get more deviations in between the contexts of the different domains. Yeah.
Okay, thanks. Yanina Panktarova has a question sort of related. So how much control is there for these designs over the size experimentally? For instance, like if you design a pentamer, do you see trimers as a suboptimal formation, or and sort of, yeah, how well defined is the geometry of the design?
Yeah, so actually, surprisingly, very often these things assemble completely on target. If they're off target, they're like lights away off target. So our classic off target is it doesn't express or it aggregates, right? But if it assembles classically, we are pretty much in the right place. We had a few examples where our interface actually decided to have a shift in terms of the helices that were interacting. And there we saw some off-target assemblies. So instead of an icosahedron, we got an octahedron. But classically, these things are pretty much on spot. One more recent example that we had actually comes to the story that I did not share, which is about structural flexibility. So once we actually introduce flexibility in the building blocks, it becomes way more challenging to guarantee that what you get is the correct outcome. And there is other things that play into account because you're no longer defining those angles really strictly. And of course, the other aspect of it is that the larger is the oligomer that you build. So instead of a trimer, if you go to something like a dodecamer, so 12 subunits, the differences from going from 11 to 12 to 13 are so small that actually getting errors in that situation is much more probable than if you are looking at a shift from a trimer to a tetramer. So it strongly depends on like the specific design problem in that sense.
That makes sense. Oliver Zerve has a question. What would be the potential application for the asymmetric assemblies that are responsive to different ligands?
Yeah, the way that we're thinking about it is sort of like a multiplexing sensing assembly. So if you would have just one of the ligands presence in the environment, then you have the formation of a dimer, and you can specifically detect that dimer. If you have two of those ligands, so potentially you have two different types of dimers, or you have a trimer forming. And so in that way, it's a way of being capable of telling what exactly is present in the environment, and in that way also sensing the environment. One application that we have been excited to explore is actually in the developmental biology, so actually in organoids, in what would happen if you use this type of assemblies to basically hijack signals from the environment, and in that way stall the development of the organoid. So how important is signal A versus signal B? Can we actually remove them one by one and see what then the effect on the development is going to be? Yeah.
Thank you. That is really interesting. I think we're going to move on to our next seminar speaker, and there's still some more questions open in the Q&A, Alena, if you're interested in answering them.
Yeah, and we can always come back to them in the end. All right, thank you. And then I guess we move on to Minkyung. Minkyung, want to take us away?
Okay. Right. Hello, everyone, and thank you for joining today's webinar. I'm Minkyung Baek from the Department of Biological Science at Seoul National University. So today I will be discussing how AI has revolutionized our ability to predict protein structures and interactions, mainly focusing on advances my colleagues and I have made in this rapidly evolving field. So we'll explore the journey from basic protein structure prediction to complex protein-protein interactions, and even recent extensions into the nucleic acid prediction. Okay. Oops. So let's start with a quick reminder of the central dogma of molecular biology. So as you already all know, the DNA is, oops, sorry. Yeah. So as you know, the DNA is transcribed into mRNA, which is then translated into the proteins, basically the workhorses of our cells. So, and proteins perform an incredible array of functions in living organisms. So they facilitate transport, act as enzymes catalyzing biological reactions, and also they participate in signal transduction pathways and form essential components of our immune system. And the function of any proteins is intimately tied to its three-dimensional structures. And these structures emerges as amino acid chains fold into complex shapes driven by various chemical interactions. So for decades, determining these structures experimentally has been challenging and time-consuming and also expensive, basically creating a bottleneck in our understanding of protein function. So I want to share one of the key concepts that really changes how we predict protein structures. So we know that protein shape determines its function. So if the structure changes too much, the protein usually cannot do its job anymore. So take a look at this myoglobin example. So in this figure, it shows myoglobin from 5 different animals. And these animals evolved separately for millions of years. But you can notice something fascinating. Basically, their myoglobin structures look almost identical. This is because they all need myoglobin to bind oxygen efficiently, and that specific function requires a very particular structures. So during the evolution, nature is quite practical. So when random mutation happens, those that mess up the protein's function get eliminated, but mutations that keep the structure working properly can stick around. And this is why functional structures tend to be preserved over time. And this leads us to a really useful insight. The proteins that share a common ancestor, what we call homologs, tend to maintain similar structures even as their sequences become quite different. So we can use these principles by collecting these related sequences and aligning them into what we call multiple sequence alignment, or MSAs. And these MSAs reveal the important structural information. So when we can see certain positions in the protein changing together across species, it usually means these positions are physically connected in the 3D structure. So for instance, if the two amino acids need to fit together in the protein core, and one changes to bigger amino acid, then the other might change to a smaller one to make some space. So basically, they compensate for each other. So basically, we can find some structural patterns from the MSA. Then the question is, how can we efficiently extract these structural patterns from the MSA? So this is where AI come into play. As illustrated in this slide, AI algorithms attempt to learn multiple levels of representations using a hierarchy of layers. And the AI is basically remarkably effective at identifying patterns or learning some hidden patterns. This is basically precisely what we need for protein structure prediction. So when provided with sufficient training examples, these AI systems begin to recognize hidden patterns in the data and respond accordingly. So for the protein structure prediction, AI can learn relationships between sequence patterns in MSA, basically the structure patterns in MSAs, and then resulting the 3D structures. So our approaches for the protein structure prediction, named Rosetta-Ford, basically utilize a 3-track attention-based network. And this the original work published in Science, and the updated version you can find the preprint on bioRxiv. And as you can see in this diagram, the Rutherford basically processes information along the parallel tracks. The first 1D track that handles the sequence information from the MSA to extract the structure patterns, and the 2D track that captures pairwise relationships between residues and structure patterns encoded in the MSA, and also the 3D track that builds and refines the actual 3D structure. And these tracks communicate with each other through the attention mechanisms, basically allowing information to flow between sequences, pairwise and 3D, the pairwise interactions and the 3D representations. And the network iteratively refines its predictions through the multiple cycles, achieving progressively more accurate structures. The effectiveness of AI-based approaches became dramatically apparent in the 14th Critical Assessment of Protein Structure Prediction, the CAS14 competition in 2020. So as shown in this graph, the AlphaFold2 from the DeepMind achieved unprecedented accuracy. So with an average GDT score close to 90, they considered roughly equivalent to experimentally determined structures, and also our Rutherford model that performed remarkably well. And this represented a genuine paradigm shift in structural biology. So for the first time, we had computational methods capable of predicting protein structures with accuracy approaching experimental methods, but at a fraction of the time and cost. And those examples illustrated the quality of predictions. The green structures are experimental results, while the blue ones are the computational predictions. Their correspondence is very remarkable. But predicting individual protein structures is only part of the story. So in the cell, proteins function in complex networks. So understanding biology requires knowing not just what does this protein look like, but also who does it interact with, and how do they interact structurally. And just like with individual proteins, the evolution leaves some signature on interacting protein pairs. So here's how. So when two proteins physically interact, certain amino acids on their surface often need to maintain the compatible physicochemical properties, such as charge, shape, and hydrophobicity, so that the binding interface remains stable over the evolutionary time. So as a result, if a residue in one protein mutates, the interacting residue in each partner protein may also mutate in a compensatory way, also known as the coevolution. And these correlated changes can be detected by comparing the sequences of interacting protein pairs across many species. So if we can align the orthologs of protein A and B from dozens or hundreds of species, and we can look for patterns of statistical coupling between residues, strong signals that these proteins interact in a conserved manner. So in other words, there are evolutionary pressure not only preserves the individual protein structures, but also preserves their interactions. So by analyzing those paired MSAs, we can extract information about whether two proteins are likely to interact, and even which residues are likely to be contact at the interface in their complex structure. So to explore protein structure prediction AI's potential for the PPI prediction, we basically developed a pipeline to predict PPIs across the entire proteomes. So a test case was the yeast interactome, where we screened tens of thousands of protein pairs. So we used the paired MSAs passed through the Rutherford or AlphaFold and predicted their interactions as well as the complex structures. And this allows us to generate a large set of high confidence interaction models, many of which were later validated experimentally. And it was one of the first large-scale demonstrations that AI can be used for proteome-wide structure-based interaction mapping. But in our yeast PPI prediction project, there was one bottleneck, which is the computation efficiency. So Rutherford was fast, but less accurate than AlphaFold2, and AlphaFold2 was accurate, but computationally very expensive. So we need to improve the Rosetta-Ford for the PPI screening to make it even faster or even have the similar computation scale, but more accurate so that we can use this as a first screening method. So we developed the RF2Lite, an optimized version of Rosetta-Ford, specifically designed for faster and more accurate PPI screening. So our previous screening pipeline employed a two-stage approach, the Rosetta-Ford 2-track for initial screening, and AlphaFold 2 more detailed analysis of promising candidates. The RF2Lite improves upon Rosetta-Ford 2-track by incorporating concepts from AlphaFold 2, such as recycling, self-distillation, and some improved inductive bias in their model architecture design. And also simultaneously, the RF2Lite is more computationally efficient than AlphaFold 2. So we achieved this by reducing the computational complexities from L cubic to L square through the structure-guided pair attention instead of triangle self-attention, and also by optimizing the network architecture. And this diagram illustrates the architecture of the RF2Lite and how it trained. So when we trained Rosetta-Ford for positive, negative PPI cases, we basically provide the paired MSA as input. And for the positive cases, we predict the histogram from the complex structure, and also we make it to predict complex structures. But for negative examples, we make it predict non-contact bin for the interchain region of the histogram, because it should not have any contact between chains if they are non-interacting pairs. And for the structural loss, basically we use the PAPE, but we masked out interchain fake loss for the negative example so that it can just put two chains in random orientation. So as you can see in this performance graph, RF2Lite shows the better screening performance than RF2-track. Basically, the larger area under this PR curve means the better screening performance. So the RF2Lite shows the better precision and also slightly better record than RF2-track, but the AlphaFold 2 still shows the best PPI screening ability. And also the RF2Lite showing much efficient computation compared to AlphaFold 2. Even though we make a little bit of advances in the architecture designs and we make it utilize more parameters, the RF2Lite version still has a similar scale to in terms of the computational efficiency, but it also shows the much faster calculation speed compared to the AlphaFold 2. So those two observations makes RF2Lite ideal as an initial screening tool, efficient enough to process millions of potential interactions, yet accurate enough to identify true positives reliably. So with this improved tool in hand, we applied RF2Lite to 19 bacterial species of interest, mainly focuses on pathogens. And the table shows the distribution of essential genes and virulence factors across these species, highlighting the scale of our analysis. So basically, our goal was to identify interactions involving these critical components, as they often represent the most promising target for therapeutic intervention. And a critical aspect of our approach is the accurate identification of orthologs to capture meaningful co-evolutionary signals. So when genes duplicate during evolution, one copy may retain the original function, including specific protein interactions, while the other copy might acquire different functions and lose certain interactions. The proper ortholog identification ensures we are capturing co-evolution between the interacting partners, the real interacting partners, rather than paralogs that may have diverged functionally. So we basically employed the reciprocal best fit algorithms to identify orthologs and then generate paired MSAs by matching orthologs from the same species. And this careful ortholog handling significantly enhances the accuracy of our predictions and is an essential step in the process. So this diagram shows our reciprocal basket algorithms for the accurate ortholog identification, which employs rigorous filtering based on both e-value and the sequence identity. And the paired MSAs are then constructed by matching orthologs from the same species, ensuring evolutionary constraints are properly captured. And our screening strategy proceeds in 3 stages here. The progressive filtering down candidate interactions. So first, we use the direct coupling analysis, the very traditional statistical method to predict the contact between protein residues to detect co-evolutionary signals at a global level, just to make sure we can do this as fast as we can. And then we apply RF to light to predict interaction likelihood and model the complexes for the most promising candidate. And finally, we use AlphaFold2 for higher resolution modeling of selected candidates. So we began with over 1 million pairs involving essential genes or virulence factors and filtered them down to just 562 high confidence interactions. And as shown in this Venn diagram, among our high confidence predictions, half of them are supported by the complex structures in PDB or string database, but the other half is newly detected ones. And we further extended this to screen an additional 3.82 million pairs involving essential proteins and biological processes of therapeutic interest, and identifying 3,050 high confidence interactions. And several of these predictions were experimentally validated using co-immunoprecipitation, as shown in this experimental result. And the high-quality structural models generated by our approach provided detailed insight into protein-protein interactions across various functional categories, enzymes, transcript and translations involving some virulence factor, and also even for the protein-protein interactions for the uncharacterized proteins. And we've also predicted numerous higher-order assemblies, complexes involving 3 or more proteins that form some functional unit. And those structural insights basically enhance our understanding of how proteins work together at the molecular level and can guide the development of new therapeutics targeting specific interactions. So deep learning has empowered a new era of structural biology, and by extending these tools from single protein structure prediction to protein-protein interactions, we are beginning to map the interaction networks that underlie microbial life and pathogenesis in this work. And our approach, combining the evolutionary analysis with the structural modeling, provides a powerful framework for discovering therapeutic targets in the ongoing battle against the infectious disease. And looking forward, one of the biggest challenges ahead is predicting cross-species interactions, such as those between host and microbiome proteins. So as shown in this slide from Ian's work, the current methods struggle with mixed eukaryotic bacterial protein pairs, the MM or ME here. And this difficulty lies in generating accurate paired MSAs across different kingdoms of life. So basically, new methods, perhaps based on some transfer learning or semi-supervised approaches, will be needed to overcome those limitations and map this complete interaction network between human hosts and their microbial inhabitant or pathogens. So let me change my gear to the protein DNA or protein RNA interaction prediction. So I'd like to discuss our most recent work extending this method to predict structures involving nucleic acids, both DNA and RNA. The RNA structures, similar to protein structure, can be inferred from the evolutionary information in MSAs. The conserved base pairing patterns provide some strong signals about RNA structure in alignment. And the idea that the RNA can show the similar structural patterns in the MSA, basically it leads us to extend Rosetta Fold for the protein nucleic acid complex structure prediction. So in collaboration with Frank DiMeo and Ryan McHugh in his group, we developed the Rutherford NA, published in Nature Method. And this model maintains the 3-track architecture of Rutherford, but extended to handle nucleic acid tokens. So we trained the model on a combination of protein-only structures, as well as the nucleic acid containing structures. So we evaluated the RFNA's modeling performance using an independent test set. So to assess the performance of RFNA, we used 2 key metrics, the aridity to measure the accuracy of the predicted structure, and p-aridity to see how confident it is for each prediction. And the results showed that while only a small fraction of predictions achieves high accuracy, RFNA can reliably identify when its predictions are likely to be correct and when it's not likely to be incorrect. And this ability to assess prediction confidence is crucial, as it allows researchers to focus on most promising models for further study or experimental validation. And when RFNA is confident about its prediction, it identifies not only where to bind, but also accurately predict their complex structures. And this RFNA often outperforms traditional protein DNA docking methods. So on the left, the dots above the diagonal means RFNA predicts more accurate structures compared to the traditional docking method, HDock. And this is because RFNA can capture the structural changes that occur upon binding, a factor often not considered in traditional docking approaches, which assume that both molecules remain rigid, as shown in those examples. However, RFNA can produce catastrophic failures, as shown here. So it's sometimes violating fundamental chemical principles like Watson-Crick base pairing, or even it shows the severe clashes. And it also has limited ability to accurately predict binding specificity. So we are currently actively addressing these limitations in ongoing research. So to conclude, AI-based approaches have truly transformed our ability to predict protein structures and interactions. The journey from single protein structures to complex interaction networks, and now to predict protein nucleic acid complexes, has provided unprecedented insights into molecular machinery of life. And recently, DeepMind released AlphaFold3, which aims to be a more general biomolecular modeling system, expanding beyond proteins to nucleic acids and small molecules. And this reflects the broader direction of our field is heading, basically comprehensive modeling of all biomolecular interactions that drive cellular processes. However, despite these impressive advances, significant challenges remain. So cross-specific interactions, such as those between host pathogen or host microbiomes or antigen antibodies, as shown here, are still particularly difficult to predict. And similarly, protein nucleic acid bind predictions and RNA structure predictions still lag behind pure protein structure prediction in accuracy. The limited training data for nucleic acid structures result in issues like structure classes and imperfect base pairing, even for AlphaFold3. So looking ahead, our lab is currently working on improving those biomolecular structure and interaction prediction using AI. And these advances will have significant implications for basic biology, drug discovery, and our understanding of disease mechanisms. So I'd like to acknowledge the many collaborators who have contributed to this work, including Frank DiMeo, David Baker, and also Chen Kong from the UT Southwestern, and many others. And I also want to thank our funding sources and also the group members who are actively working on advancing the computational tools for predicting and designing the biomolecular interactions. And thanks for your attention. And I'd be happy to take any question you may have.
Thank you, Minkyung. This is super exciting. Do we have questions? I think we already have a few questions in the chat. So Matthias Bock is asking about the type of MSA that you get in the case of pathogen-host interactions. So he says that one would presume the paired MSA is asymmetric, as the host is trying to escape the pathogen in evolution, while the pathogen is trying to outfox or keep up with the host. And so is this something that you actually see in the MSA evolution that is different from like normal PPIs, that actually both sides are trying to keep up with each other? Or is there no signal at all in the MSAs?
That's a very good question. So so far we actually don't know whether it has the same PPI patterns or it might have different patterns in their MSAs. But one thing, because host tries to escape from the pathogen infection and pathogen tries to impact the host, is also kind of co-evolution. So there should be some kind of patterns in the MSA, but we are not sure whether it will be the same patterns in normal PPIs or it may have different the patterns for these host pathogen cases.
Yeah, maybe from my side, like sort of related, we've been seeing that a lot of these methods are also not particularly great in predicting this type of assembly PPIs that we typically have, that are highly cooperative and actually are on the weak side of protein-protein interactions. What are your thoughts there? What could be the pathology, or what could be improved to go towards that direction?
So as the one who actually measured the physical chemistry, I'm always think the AI needs to run some physical principles to predict the general protein-protein interactions and also all the assembly problems. But the key question I have, whether we have enough, structural data to train that kind of model which can understand the general interactions. So I think the AlphaFold3 and also the Rosetta-Fold or Atom, I didn't mention it in the presentation, but those general biomolecular interaction prediction methods are actually the way toward understanding the physical principles upon binding by training the model based on the general biomolecular interactions, which governed by the physical rules. So I hope this kind of approach, utilizing more general biomolecular interactions, and also tries to augment the data, which can tell us how the proteins or how molecules interact to each other, that can help us to identify weak PPIs as well as those actually the cross species or antigen antibody cases as well.
Yeah, I think in that direction, there's also questions about how would you treat when you have intrinsically disordered regions participating in the PPI, or if you have multiple conformations that are connected to the PPI. How do you go about there with the MSAs and in your design of your pipeline?
So for the intrinsically disordered region, so actually at this moment, we basically ignoring them, or we intendedly do not pay attention on those regions, because what in our benchmarks, the AlphaFold and Rosetta-Fold is somewhat good at finding the interactions or the where to bind, the binding pocket on the partner protein, but the motif region on the IDRs or the correct identifying correct the interaction mode or the complex structure are still very challenging. So in current work, we mainly focuses on structured region, but yeah, IDRs are where we tries to improve our our method, but it's a little bit difficult at this moment.
Cool. Well, I'm sure there's going to be more discussion later on. So thank you so much, Minkyung, for this awesome insight. And we move on to Bruno. Take us away.
All right, let's see if we can make this work. Well, the news are we can't make this work. All right. How's this looking?
Looks good. Looks good.
You see the right screen? You see the right screen. All right, well,
Yes.
so thanks a lot, Alena, for organizing. Thanks a lot, the Protein Society, for continue having these type of gatherings, even if virtual. So today, rather than telling you about 30 things that we do in the lab, I decided to just tell you about one and actually spending time thinking about this problem. And it's a really, for me at least, it's a really good segue on Minkyung's talk, because I guess there's a lot of conversation about, or there's a lot of doubts in our minds about what these models actually learn, and you know, using them for design is in fact one way to try to understand what they actually can capture and what they actually cannot capture. So I'm going to spend the entirety of my talk telling you about our thoughts about what has worked for us, what has not worked for us. You know, generally, like everybody else, I will focus mostly on what worked and not necessarily on what has not worked. That's like, you know, it's a common pathology. But today it's really sort of like, you know, I think the question for us is really to just think about, well, you know, this de novo design of protein-protein interactions has really been a very hard problem in computational design, one that I remember very clearly since when I started in this field, that we just seem to have no way to get at this. And I think more and more as we see the different contributions coming out, it starts feeling more as a solved problem, although we need to be extremely careful when we label something as a solved problem, because what is solved for some people might not be necessarily be solved for others, and because there's sort of like a number of different aspects and nuances of this problem which clearly are not solved. But before going into business, I think, you know, every time that I'm involved in a Protein Society event, there's always a few memories that come into my mind. And so I guess my connection to the Protein Society has been there for, I guess, more than 20 years now as an undergraduate student. And I guess I'm super proud always of thinking about the first paper that I ever published as an undergraduate. And so you can see sort of like, you know, this sick amyloid fibrils that by the time we were modeling in the computer, and really this, for me at that time, felt like, you know, unreal. And I think right now, sort of like with the kind of things that we can think about, is really, really interesting to see how far we came. And then, of course, you know, like I've done one of these, I'll just tell you like the funny story. I've done one of these online webinars before, and you can see here, like, you know, who are my illustrious partners when I last gathered, and sort of like an event has happened, and essentially, you know, my daughter sort of like bombed the whole thing, and essentially after I gave the talk, nobody really remembered anything that I said, but they did remember my daughter. So I guess this ultimately was an attestament for my capability to multitask, and you know, and also made her famous in some way. So really, thank you, thanks again for having me here. And let me tell you then about what I want to tell you. And today, it's really about designing protein-protein interactions. So Minkyung already sort of like gave you the whole necessary introduction, why this is such a hard problem, even in a prediction standpoint, because it's just really so hard if you don't have good data sets or good data inputs to figure out where two proteins bind together. And the same thing has been true on the world of design, that it has been extremely hard to capture what are the principles that are necessary in order to generate structures and sequences that can bind to target proteins on defined sites with predicted structures. And part of the problem And here I'm just laying out some of the issues that are difficult about this problem, is that it just takes a few interactions in order to make a protein stick to each other. That's what it turns out, right? It's not really the same type of problem that when you have a folding within a particular sequence, and that many residues intricate and basically define the three-dimensional structure of the protein, but it's rather just a few side chains at the surface. In some interfaces more than others. And here this really becomes a huge challenge for computational methods to discriminate what's a real protein-protein interaction and what's not a real protein-protein interaction. And even from a lot of the things that Minkyung has told you, you really rely heavily on evolutionary information in most of the cases. And then, of course, there's a number of different approximations that were taken, and part of it is because we really ignore a large fraction of the physical actors of this system, which are related to the solvent, and also some of the dynamics that is present in many of these polypeptides, which we continuously try to ignore, but that at some point we will no longer be able to ignore because they will be important for the problems that we're interested in designing for. So where did we stand before deep learning tools were available? And really where we stood was mostly related to this aspect that, if we wanted to design, to computationally design something, we actually have to test, 100,000 to 10,000 proteins, more like this, like astronomic numbers in some type of display platform where we could actually screen our designs. This, of course, has changed, and now with a lot of these prediction networks that can be repurposed both for generation as well as for filtering, and I'll be touching a lot in these two topics on how they've been used in different ways. We have now the possibility to actually create these binders in different ways. So let me tell you how we went about this, right? So we have been doing a lot of work here in the lab, which basically relies on using AlphaFold 2 mostly as a tool for generating new sequences and structures. And what happens here is that ultimately the way that you do this, you do this through relying on the backpropagation algorithm, which then gets optimized for particular confidence metrics, which ultimately the way that it does, you start from a random sequence, and as you start sort of like trying different sequence options, you eventually converge in solutions that are favorable according to your confidence metrics in this case. And that's really the core of what I'm going to tell you. And so with this we have used this for a number of different exercises, particularly for the design of new folds. But here we really wanted to explore how well this could work for the design of new binders, and again also get to this question of did these algorithms learn some sort of physical principles that can help us to engineer the novo protein interactions. And so, and what we came up with, or better, what Martin and Leonard came up with, is what they have called the Bindcraft, which is a name that is very entertaining on its own. So here also part of this work was done in collaboration with Sergei Ovshinnikov, and you can see here, just so that you can visualize how the process goes, and it's essentially an hallucination process, if you will, where you just provide a starting point, a random sequence to the algorithm and a target protein, and essentially as we go along the gradient of the backpropagation, we can then converge into sequences and structures that have high confidence according to this to the network that we're using. So of course that this is the computational tool. Then what we set out to do is to test, well, let's see how this works in different protein targets, because again, nothing really just is sufficient to be tested in the computer. So you see here a number of different proteins for which we designed binders. I will tell you a little bit about a few of them and highlight some of the lessons that we take from from targeting these proteins. But ultimately, you can see that we designed a number of different folds to engage these targets. As you can see, there's also there's a relatively common thread into many of them. They are most of the times heavily relying on alpha helical structures. There's of course a reason for that. It's because, you know, in a way it's easier to optimize for local secondary structures, if you will, than there is to optimize for either loops or distal secondary structures. And also that, you know, one of the features that arises from this exercise is that in fact we see these protein structures molding extremely well to the target that they're supposed to bind to. And of course, you know, here this is clearly capturing one of the features of natural protein-protein interactions, which is shape complementarity. So in that case, you know, good things are clearly happening in these hallucination processes. Now, we did express and purify a number of different proteins. So here I'm just giving you some snapshots of the experimental data that we collected, and in a more summarized way, you can see it all here again. And I'm going to, you know, ultimately give you the punchlines from this slide. And so what you can take a look here in the top is that the type of success rates that we're having are extremely high. So oftentimes we could get success rates from 10% to 50% in terms of the designs that we would try experimentally, that they would be classified as binders, but also in terms of the types of affinities that we're having. So these affinities that we have here are essentially affinities that are coming out of the computer. So there's no experimental optimization, and you can see that they go anywhere from dozens of nanomolar to low micromolar. Okay, so then I guess now the new or some embodiment of what CASP used to be is these kinds of competitions for binding. And so, you know, we were lucky that our neighbors here in Lausanne were running one of these competitions. So Martin and Leonard went for it and sort of like designed a binder that was able to win this competition. And funny enough, the second place had been a summer student that we have had in the lab that has used a version that he has developed himself, which was very much relying on some of the things that he has done here in the lab. So that was kind of like a very interesting type of validation that it was unbiased. And so that was sort of like, you know, a big victory for Conde that on its own got like, you know, a wonderful second place. But so one of the things that we wanted to try, and this is always one of the questions, and I will come back to this question later on, because one of the things that I'm very interested in doing during the next 10 or 12 minutes that I still have left is to try to understand what actually these algorithms do and how well do they generalize, or if they in fact, whenever they start hallucinating binders, the only thing that they do is to actually create three-dimensional motifs of protein interactions that we already know. And so in order to test that, what we did was to basically just take a de novo protein, which essentially didn't really exist in nature, and just try to design binders to it. So, you know, we took this beta barrel that we have designed for a completely different thing and designed you know, equally funky protein. As you can see here, this does not strike me as the kind of design that we could do earlier on. However, when we actually check the affinity of this binding event, you know, it really sort of like looks pretty good in the SPR. And then we get a crystal structure, and again, sort of like, you know, the binding mode that we had predicted in the computer is very closely recapitulated. So now, you know, this was great, and you know, like we started sort of like thinking about what we could do with these binders. And here, you know, as in any type of engineering exercise, very much the limits are your imagination. So your imagination could either be your best friend or your worst enemy if you don't put it under control. So I'll just sort of like tell you a little bit about the few things that we did. And Leonard and Martin started to get very excited about, for instance, targeting allergens. And so here you go, sort of like two examples of two binders, the models, again, sort of like their affinity curves and the crystal structures, again, show you that we have bind with very high accuracy to this target. So you also can start seeing that, you know, again, the types of binding modes that we are not necessarily, although the proteins are made with helices, they aren't necessarily just like helical motifs binding to the surface of the other proteins, but rather having like a full engagement of the fold with the target protein. Then we started doing a few things more complicated, and you know, here Martin sort of like went to try to design proteins that could in principle control gene editing proteins, another one, so like the case of Cas9. And so here the idea was that we could in fact bind proteins that could compete the DNA binding interaction. And so we targeted this REC1 domain, and what you see there is again this protein sort of like being generated, coming out of this large pocket that it has, and completely molding into the DNA binding site. And here we got only a few low resolution reconstructions on EM, but that essentially tells us that the designs are at least binding in the right place, given that these are actually pretty large proteins, but we don't really have high resolution structures for these designs. And ultimately, we were able to show in collaboration with Gerald Schwank and Lucas Kissling that these proteins do in fact inhibit the gene editing capability of Cas9 proteins. Then one of the things that I'm really interested is about the complexity and how can we actually use these binders in order to modulate biological phenomena that are not that go beyond binding, that go beyond a protein just sticking to one another. And one of the things that we got excited about was how could we actually repurpose using these binders, these AAVs, which are typically used or are the vector of choice used for gene delivery, and how could we actually repurpose them to actually transduce their cargo in specific cell types rather than other off-target cell types. And so the idea here is that if we actually generate the binder that recognizes specifically the surface markers of the target cells, then this in principle will avoid the transduction of the genes that you don't want in unwanted cells. So just showing you some of our brief examples here, a structural representation of how these capsids, after being engineered with the mini binders, could look like. And one example on how we actually started targeting cells expressing, for instance, this immune receptor, in this case PDL1, and how we could actually see that 2 of the designs out of the 8 that we've tried, in fact, do transduce in their target cells and don't transduce in a cell line that is expressing some other random receptor. And so again, and this for us was very interesting because we went straight from design to transduction, or to function, if you will, rather than characterizing the proteins first. So we expressed this AAVs together with our neighbor Bernard Schneider, and we took these supernatants and just checked, do you actually transduce or you don't transduce? And this for us was a very interesting exercise of trying to see if in fact these binders could be useful for something else rather than just binding proteins. Okay, so but something that I typically like to ask myself and also the people in my group is, well, since this seems to kind of work and with relatively high success rates, pretty much comparable to the standard of the field, which I guess one could say that it would be RF diffusion. Well, so what just really happened here, right? And I think one of the things that we started sort of like looking was, well, how many design trajectories do we actually have to run in order to have, let's say, 100 successful designs? So, to give you an idea on how this pipeline works, you start doing your hallucination process, you get a bunch of different filters after the sequence is generated, which are both based in AlphaFold as well as in physics basic metrics like hydrogen bonds, surface areas, all these kinds of things. And how many trajectories do we actually need to run in order to get to 100 successful designs that pass all the filters? And so you see that it varies from target to target. Some targets you only need, let's say, 300 trajectories. Some other targets you need a lot more than that, and this again is inherent to probably properties of the target itself, on how bindable it is, this target, if this is even a word that exists. Now, again, the conversation here is that, oftentimes what we hear, and this is, it is true, that this is a procedure or a protocol which is computationally very heavy, because it relies heavily on AlphaFold, which is a large network. And so as we start working with bigger proteins, it doesn't scale very well. And that's basically what you can see here, right? So as we start sort of like hallucinating higher or larger proteins, you see that the trajectory time just goes nuts, right? So ultimately, you go from a few minutes into dozens of minutes, and now if you have to sample many trajectories, this will become very difficult to manage in the computational side. On the other side, if you go just for straight up RF diffusion trajectories, this is extremely fast because it relies on diffusion, and as such, it scales extremely well. However, something that it's not as often discussed is that whenever you use RF diffusion, and this was also something that has inspired the developments that we have done here, you are, of course, heavily relying on a filtering procedure, which basically relies on AlphaFold. So for each one of your designs, you actually have to repredict everything. And so if you actually take into account the full generation, which is very quick, obviously, but then you also have to continue repredict everything. In fact, when we compare the time that it takes for us to generate a protein and the time that it takes for RF diffusion to generate proteins, then things are much more balanced. And so you can see more or less here, just by comparing the different bars. And again, they become more balanced because they're actually relying on the same thing. The critical difference here is that what we did was, rather than using the oracle as a filtering tool, we actually use the oracle as a generation tool, because since the oracle seems to behave so well, then we might as well just use it. Okay, now what other things do we see that are happening in this algorithm that make us confident that tries us to explain a bit why do we have these success rates? And part of it is also due to the type of amino acids that we get or residues that we have in the interfaces of the design. So we see a lot of interesting large hydrophobic amino acids showing up at the interface, which, for instance, if we just compare with RF diffusion designs, that's not the case, right? And again, here we also see, for instance, one of these phenotypes that is very common in problematic designs, which is we have more alanines in many of these designs generating by RF diffusion than by Minecraft. And so this also helps to explain a little bit more why some of these designs might actually work very well. Then to the question of are these interfaces actually novel? And so I'm going to take a few minutes to unpack this plot. And so what you see here is that essentially many of the folds that we generate are not necessarily new. Many of them are actually simple helical bundles. Life is great. So you do the TM score, you compare it to other folds. Many of them have been seen in nature, and that is okay. Now, when you only focus this structural analysis in terms of similarity to other interfaces that you find in nature, what you see is that, in fact, the TM scores that are reported are extremely low, which basically means that we are not just recreating interfaces that have been seen before, but we're in fact generating new structural arrangements and new sequence arrangements that were not present in the database. And that, of course, speaks to the capability of generalization of these networks, and perhaps gives us some lights about what they actually have learned. And then, more on this pink and salmon bars, you can see that the sequence identities that are generated are just in generally very, very low. Okay, so perhaps one important point is that also in many other pipelines, in fact, the target is not given the freedom to move. In our case, in fact, we do allow the target to move. So there is some flexibility on the target. And so you can see sort of like things that are reminiscent of some type of induced fit or just repack. And you see that in depending on also the target. So here we're showing you all the targets that we've done. Some of them do drift a significant amount, at least at the interface level. So you see like 2 angstroms, 2.5 angstroms. Some of them also don't drift at all. But here there is at least this flexibility for the target to adapt. And we also think that this is contributing for the success of this design pipeline. Okay, so I'm just going to sort of like wrap it up and giving you some of the key points to take home. And really here what we've done was to use AF2 rather as an oracle, but to repurpose it as being the generator. What we observe in many of these designs, and we think this is really sort of like something that it makes the difference compared to other things that we've been trying throughout the years, is that because we have this freedom to shape the proteins to their target, this shape complementarity makes a difference. We also observe that there is a balance between the composition of the interfaces, which was a typical problem of non-deep learning approaches, where now we have like this much more balanced polar and apolar compositions. By allowing flexibility on the target, we also think that this will generally help. And many of the proteins that we generate are still helical, and this is not necessarily something that we were very surprised by, but it is also something that it's relatively easy to tweak within the algorithm. Maybe perhaps the most important point is that the intuition that we have built in the results that we've seen is that, in fact, the success rates also depend on the freedom of the algorithm to pick the folds that it wants to design and the site that it wants to engage. So there's certainly some sites that are more bindable than others, and the more freedom we give, the more likely we seem to be in terms of designing successful binders. Okay, so with this, I am not exactly sure if I'm too long or too short. I think I'm too long. But just a big thank you to all the people and that have done the work, and I'm truly lucky on having had the chance to work with the amount of like passionate people that I've done through the years. So I'm happy to take any questions, and sorry if I took too much time.
Thank you, Bruno. As always, very insightful. I love the analysis altogether. And maybe just one question so that we give enough time to Hannah. So both Finton and Lisa Fan ask, because you design relatively small binders. Sorry, I cited the wrong people, Daniel Hardy and Shlomo Yakir. They both ask, what about the not just the affinity, but also the targeting of your binders, how specific they are in comparison to off-target binding?
Yeah, so, actually that was part of the data that I didn't quite show. The best we could do, well, there's many things one can do, right? But the best we could do was to essentially just test all our binders against other targets that we had in the lab. And we had not seen any cross-reactivity towards the other proteins that we had in hand. Now, is this the most comprehensive experiment one could do? Not really, right? Because perhaps you could actually just make a pull down with some cell lysate and see what would stick. We haven't done that, but in a way, we also many of these proteins were actually kind of similar, some of them, so they had similar folds. So this is also a high bar, and in fact, what we see is that they do seem to be, in fact, pretty specific. They don't seem to be sort of like just sticky because they're sticky.
Cool. Well, let's leave the rest of the discussion for after Hannah's talk. And yeah, Hannah, take us away.
Let's see. Can you see my slides?
Yes.
Okay,
Yeah.
great.
That's perfect.
I want to thank Alena and the Protein Society so much for inviting me. This has been really fantastic to listen to all these other wonderful talks. And so I'm very excited to tell you about some things near and dear to my heart relating to protein dynamics. So something that has just fascinated me in general since the start of my scientific career is that for biomolecules, including proteins, to operate in life, many of them are machines. And sort of by definition, machines have multiple states that they have to interconvert between. And so what amazes me is that there are systems in life where even if we go down to the size of a single protein domain, and even if we do reductionist in vitro experiments, in the absence of other things, in the absence of other binding partners, what people find is that these proteins have evolved so that even in the absence of those other things, they themselves interconvert between biologically relevant states. And so here I'm just showing two kind of classic examples of this by this point. So adenylate kinase is an enzyme that is essential for life, and its rate limiting step for its catalysis is actually governed by this open close motion of these ATP and AMP lids. And this is what's rate limiting for the enzyme, but actually in the free state of the enzyme, those motions are also taking place. And so another kind of classic example by this point is this NTRC receiver domain, which is involved in cell signaling, and it gets phosphorylated, and there's a conformational change that the phosphorylated form it adopts a particular state, but what people observed is that even prior to it getting phosphorylated, it is also interconverting between those two states. And so in this day and age, what's been really interesting post AlphaFold, so this idea of wanting to predict multiple conformations is viewed as the next grand challenge. And there's all sorts of methods that come out. And this is by no means a complete list. I think it feels almost every day on bioRxiv, there's another one that could be added to this list of people wanting to use principles from deep learning to be able to model and predict these multiple conformations. And so I am on this list as well. So developed this AF cluster method that used a principle for fold switching families to use AlphaFold to predict multiple states. But before developing a method further, I had a question that I was very bothered I didn't know the answer to as a postdoc in this field who wanted to be trying to develop methods to predict multiple states. And the question I didn't have a good answer to was what I might say very tongue-in-cheek, but is there a ground truth for protein dynamics? And so what I mean by that is, from my point of view, AlphaFold was able to be an amazing breakthrough because communities had established what it meant for a model to do well at predicting protein structures. That was in the context of CASP. And also communities had come together in the context of the PDB to make large data sets of really well understood data. And so I sort of felt if some aliens came down in a spaceship and just handed me an algorithm saying, oh, here, we figured out protein dynamics, here you go, I wouldn't know what would be the experiment on a blind system that I would want to do to know if that had recapitulated this. And I'll say by dynamics, I mean multiple conformations as well as the time that it takes those conformations to interconvert. And so when I asked Dorothy Kern this, so in the Kern lab, we use all three of these structural biology methods to understand how dynamics lead to function, because all three of these methods reflect multiple conformations. But when I had this question, is there a ground truth for dynamics and time, and what experiment would I want to look for to know what type of dynamics is happening? She encouraged me to spend some time with this guy at the bottom of the pool down here. Because if you look close, he's actually smiling, because NMR has experiments that have been under development for a good 50 years by this point that actually directly measure dynamics. So X-ray and cryo-EM absolutely report multiple conformations as well, but there it's a question of deconvolving signal, whereas the NMR experiments I'm going to describe give us actually a positive indication that there is dynamics there, and that was really attractive to me. And so what is one of those? So here's going to be a bit of a crash course on some NMR concepts. So one thing that is hard for studying time in biology is there's a wide range of timescales going on. And so all the way from picoseconds to hours. From my point of view, molecular dynamics is a method that if one has questions about nanosecond timescale motions, is actually quite well poised to capture those. But past nanoseconds, past microseconds, there's a lot of biologically relevant processes that are just simply too slow for current MD methods. And so in particular, there's an observable from NMR called REX that reports fundamentally if an atom is experiencing motion at this micro to millisecond timescale. And so as an example of this, in that NTRC domain, prior to knowing what the identities of both of these states were, the way that people realized that this NTRC in its apo form was interconverting between these two is one could do one of these NMR experiments that measures REX. And so that REX, you don't get a structure out of it, you get a number per residue. And in the case of NTRC, if you then plot the values of those numbers on a structure that you know, you can see that this whole side of the protein lights up with that indication that there's a millisecond process there. And in some ways, that's a real boon, because then it takes more complex modeling to figure out what the minor state is. But this REX just tells you that there is a millisecond process there. And so as I was becoming more familiar with this field, I was realizing that there are a lot of these REX measurements in the literature. People have been doing this for 30 years. But there was no unified collection of them. And as a data scientist, you know, to make advances in things, we need benchmarks. And so there are a lot of these colors on structures in the literature. And some questions that I then had was, well, one, how much of this could I collect and put into a benchmark? If I did that, could I learn something interesting about how dynamics and evolution are related? That's ultimately a question that drives me. And then also, can existing models already predict this if we can gather this up and standardize it? And so crash course on how these measurements come to be. So step one of any NMR experiment is one has to, quote unquote, assign their protein. So one has to sort of figure out where in chemical space each residue falls. So each residue in one of these bread and butter HSQC spectra, each residue becomes one peak. So after you've done that, after you know where your protein lies in chemical shift space, what one then does is a relaxation experiment where, so here I'm zoomed in on my protein, and if I'm looking really close, because I've labeled it with isotopically active nitrogen, all the little spins are pointing in the same direction in my magnet. And I'm then going to hit it with a 90-degree pulse, which is equivalent to if I had a top spinning on a table, and then I knock the top over, and the top is going to continue to precess on the table. And depending on what friction the top feels, and all these little tops are in different chemical environments, so they're going to feel different types of friction depending on their environment surrounding them, they will relax at different times and return to equilibrium at different times. And so people figured out the quantum and the equations of how to then model different timescales of different processes in those relaxation experiments. And I'm just boxing here broadly the terms. So from the type of data that is most widely available, one can observe motions at two different timescales. So this S squared, this backbone order parameter, can be determined, which corresponds to amplitudes at the nanosecond timescale. And then also this REX can be determined that relates to where there's millisecond motions. And so here's another way of looking at that data for that NTRC domain. It's common to plot the R2 over R1 ratio, which sort of cancels out any contributions from nanosecond motion and tells you where there's spikes in this ratio is where there's REX due to these millisecond processes. And so how many of these data sets can one postdoc curate before she gets tired of this endeavor? The answer is 133. And so roughly half of these came from the home of other types of NMR deposition. So the BMRB is where chemical shifts are required to be deposited if they correspond to a structure. There are currently no deposition requirements related to relaxation data, but there's been some number of brave souls volunteering to deposit their data there anyway. And the other half came from me sort of trawling the internet and emailing people, and then also doing this masochistic work of plot digitizing some classic data sets from the 90s that I wanted to include. And so here's then an example for each of these 133 proteins. So these are all in their apo form, no homodimers, unfortunately. We wanted to keep those 133 as simple as possible so that any motions that are present are truly there intrinsically without the due to other things binding. And so then these two measures tell us about if there's nanosecond motion, millisecond motion, or nothing. And so this is just indicating this for an example of this calmodulin domain. And I want to show people who maybe aren't familiar with this type of data, this plot in particular. I don't claim to say I know anything about calmodulin, but so this het NOE tells us where there's nanosecond motion. Here in calmodulin, there's nanosecond motion in these loops, but this R2 over R1, this REX measure, it shows us that there's other processes that is being experienced here in these helices. So there really do exist these measurements that tell us what motions are occurring at different time scales. And so I put together this benchmark. If you make a benchmark, you have to give it a name. So I named this RelaxDB. And so here's an example of a handful of the proteins that are now encompassed in this benchmark, and just showing a variety of, there were some technical things that go on when things are highly anisotropic to make sense of the data, but we were able to develop a robust pipeline to accurately fit all of these at scale. And so with that data in hand, of course, 133 is not enough data to train a new model from, but I was curious if there are any trends that we could observe with that data all in one place. And so one thing that I was really struck by, so one of the first things that I tried doing was I wanted to calculate sequence conservation for each of those residues in all those 133 proteins. And if I did that, what I observed, in particular in residues that are buried in the structure, residues that had millisecond motions through this label from these relaxation experiments, those residues were the most conserved. They were more conserved than, here in gray, were residues with no motion label of any sort. Those were also then more conserved than residues with nanosecond timescale motion. And what this speaks to is that residues with millisecond motion are often related to the function of a protein. It's been interesting showing this plot to different people. Some people are very surprised by this. Maybe our intuition might have been that the structured elements would be most conserved. But then to other people, this makes total sense, because they say, oh, well, my protein that I study has this allosteric pathway, and that's related to its function. And surely that definitely is a millisecond process, and it's also definitely highly conserved. So I think this is important to see, because as we're going to have continuing on bioRxiv every week, more and more models wanting to predict dynamics, there's going to be different evolutionary sequence patterns underlying different timescales. So another thing that we then realized about this data set was that it didn't include all the information that we could have about millisecond motions, which is really this regime that I was interested in. Can we predictively model? And so the reason for this has to do with that first step in doing an NMR experiment, where one is assigning one's residues. So some of the residues never even appeared in a relaxation experiment for this dirty secret of NMR, which is that if a residue is interconverting between two different states, depending on the chemical shift difference between those two states and the time scale of that interconversion, there's a variety of things that can happen. And so there's a bit of an analog with visual things as well. So if I have a stoplight and it's flashing between red and green, if it's slow enough, I'll observe it as two different peaks. And so that's the case that what happens in an NMR spectrum if there's a very slow interconversion. If there's a fast interconversion, the red and green just average it's going too quick for my eye to distinguish them, and they'll blur into an average color in the middle. But there's this danger zone where what actually happens in the spectrum, if the conversion process is just at this intermediate time scale, the peak can broaden beyond detection. And so a number of NMR spectroscopists, when I talk to them, they say, actually, the most interesting parts of my protein I can't see because they've been broadened beyond detection. And so Doro, my postdoc advisor, pointed this out, and she said, well, Hannah, you should go back to your 133 proteins and find all the assignments and figure out, for the ones that didn't make it through the relaxation processing, which of them are missing due to other, artifacts not being able to fit the exponentials correctly or whatever, and how many of them are missing because they weren't in the assignments. So I did that, and it was really annoying, but we got it. We did that curation, and what was really interesting to see was that the residues that had missing assignments in the amides followed the same patterns of being more conserved than these other categories in the core residues. And so it was kind of a painful curation experience. And so then I was sitting in Dora's office and just realized, wait a minute, if a signal that we care about could be these missing amide assignments, wait a minute, we actually have that data for basically any protein anyone's ever done NMR on, not just those specialized relaxation experiments. And so what the vision then that I had was can we think about training a model to predict millisecond motion by just saying, can we create a model to predict what is missing from the BMRB? So I did some data curation things to remove NMR experiments done in conditions where we can't, there's other reasons why peaks might be missing for a variety of labeling or other technical reasons. And so, but after doing that cleaning, still we had 9,000 proteins for which we could create this binary label of which residues are present and then which have their backbone amide missing. And so we're making the assumption that if something is missing, it is exchange broadened. And so after doing that, what was interesting to see is that even for these, under 200, many of these are under length 100, there are actually a lot that have missing assignments. And the nature of those residues with missing assignments was quite different than what is missing from other types of structured data. So this is an example of this that I wanted to get some statistics on. So here's a MAP kinase binding domain, where here in red are the residues that are missing in the NMR spectrum of this protein. But if you solve a crystal structure, the residues over here are missing in this loop. And you can see they're different subsets. And so our hypothesis is that, or our intuition is that things that are missing from NMR represents millisecond motions, and things possibly missing in X-ray or EM density are possibly nanosecond or other processes. And so what I saw as well for the subset of proteins for which I could reliably compare NMR spectra and X-ray EM structures, that there's very little overlap between residues missing in NMR and then also missing in X-ray. And the B factors between what is assigned or unassigned, it's statistically significant, but there's very little difference in these distributions. So these missing assignments and these B factors are really reporting on different things. And so it was at this point that Gina El-Nesser, who's finishing her PhD in Posu Wang's lab, got interested in this project. And I was really happy because there's all this data and so many deep learning questions to be asked. And so we had this question then, can we train a model to predict which peaks are missing? And the hypothesis then is that a model that does well at that might have actually learned something about millisecond motions. And so we can pose this then as a very simple binary classification problem. Our data splitting was we combined the missing BMRB, the relaxed DB, and a set of another 10 that I had curated from CPMG, more high-powered NMR experiments. We did the standard thing of clustering these to 80% sequence identity and used these to then split off a training, a validation, and a test set comprising of both the missing peaks data, but then also this much more nuanced relaxed DB data. And our initial screening we did with only a 80% sequence identity split. But if you have it all done machine learning, you might kind of be screaming at seeing if that would be our final model. And that is not our final model, but this question of training data set identity is actually quite interesting for this problem. So this is a new task. So this means we need to start with some baselines as to how baseline models can do at predicting missing peaks. So the baselines we started with is kind of a dummy classifier. So there's certain likelihoods just out of statistics from the training set. For instance, loops with histidines and loops are always missing 14% of the time. So I could make a dummy classifier that just says, if this is a histidine in a loop, 14% of the time I'm going to say that it's missing and the other time not. So that dummy classifier doesn't do much better than baseline, if at all, for considering amino acid type, secondary structure, and then also jointly amino acid and secondary structure type. So another baseline for our final model is, let's say I just take my amino acid, my sequence, and I put it in a vanilla 1D transformer, going from sequence to predicting this binary classification. So that does better than these other baselines. So Ramith was a very talented post-bac in Sergei Ovchinnikov's lab. He had the idea to use the AlphaFold pair representation to see if it had predictive power for this task. So he designed this architecture where we're using AlphaFold pair representations, no MSA, like just to understand AlphaFold structure-based element, and developed this method then to go from the pair representation and do a lightweight attention thing to get out a 1D vector to do our prediction. So that did better than all these other baselines. That was really exciting to see. And so concurrently with what Ramith was doing with developing this architecture, I had been exploring how ESM layers do at predicting this. And what was quite interesting to see was that the simplest ESM architecture of just taking the last layer, putting it in a vanilla transformer, did better than the AlphaFold pair representation that we've been very excited about. So Gina was incredibly helpful for also querying all these layers in ESM2. And then when ESM3 came out, she was really interested in seeing how ESM3 did. So ESM3 is a multimodal language model. So it was trained on both sequence and structure. And so you can put in both sequence and structure to it. And so what she found was that we got the best performance from an intermediate layer of ESM3 when also providing structure to it. And so the best model we selected on AUPRC, but AUROC is more intuitive to me. So in our validation set, we saw that we could achieve an AUROC of 0.75 for missing peaks in NMR. And so this is kind of bananas to me. Like this has never existed before, so that we now have some predictive power for predicting missing peaks in NMR with an AUROC of 0.75. And so that was with an 80% sequence identity trained test split. And so, you know, this is something that could potentially allow for a lot of data leakage. We made the training set more stringent, down to 30% sequence identity, and also removed structure homologs by TM score. When we did that, the size of our training data went down by a lot, but the AUROC overall only went from about 0.75 to 0.74. So that indicates to me, one, that this is a hard problem, and that even giving all this data to the models we tested did not result in the performance getting that much better. And two, it means that the ESM models have a lot of predictive power already that was sort of primed to be good at this task, even with not that much data. So what do these predictions look like? So this is this funky classifier, which we had to give a name. So we named it Dyna1. And what we get then per residue is 1p missing. So because fundamentally that's what it's trying to predict, but we interpret this as where is their millisecond motion. And so we've converged on this putty representation, or as some people call it, these sausage plots. So where a fat sausage represents a high probability of there being millisecond motion there. And so if I just take an example that did best from this test set, so again, split out by 30% identity, no structure homologs. So here's this ribosomal protein, and the Dyna1 probability is indeed higher where there were exchange broadened peaks in this spectrum. Here's another example where the motion that's there has some biological relevance. So this is a ribonuclease toxin where there's a millisecond motion in the allosteric binding site of an inhibitor that binds there. And this flexibility allows this particular ribonuclease to accept a variety of mRNAs as substrates for its cleaving. But something funny about this data was that we realized in looking at it, so here's something that could be a bunch of false positives of where there's this fat sausage, but it's gray, so there's not labels there. But when I went and read the paper, the authors commented that there actually was a lot of exchange broadening here. And so this reminds us that this missing BMRB data itself is an incomplete view of the dynamics that are there. And so we had to go to more or more specialized data set, which was this relaxed GB data. And so the real kicker test then for all of these models that we tested was, can these deep learning models have predictive power for where there's REX? So not just where peaks are missing, but where the peaks are actually present, but have measured millisecond motion. And so what we found was that they do. So this norm AUPRC is greater than 0, which indicates that they do. And so that was really exciting to us, because it means that they have generalized something about millisecond motion that isn't just due to who knows what potential chemical quirks about what might go missing. And so what was also interesting to see is that this model Dyno1 has the most predictive power in areas where there's conserved patterns of millisecond motion. So here's just some examples of this. So there's millisecond motion around the active site of this mercuric transport protein. The whole point of the paper that this YCQA flavodoxin is from is that there were millisecond motions in the binding site for where it docks its small molecule. Here's another example of this export signal that has this concerted millisecond motion. And if we looked systematically by just binning residues based on their location and conservation, and then calculating AUROC within those bins, we saw that Dyno1 did best at residues that were the most conserved. And what was interesting too was I wanted to go and see where in our data set, in our relaxed DB, what's up. There were some proteins where Dyno1 had an AUROC less than 0.5. So what was going on with those? And it took a lot of just data exploration, but a trend that I eventually realized was that a number of these proteins, and so all these points in brown, are proteins that in part of their function, they bind phosphates. So DNA binding proteins, RNA binding proteins, signaling proteins. So they bind phosphates, but they're characterized in phosphate buffer. So this is going to be a common practice if you have a lot of positive charges on the surface of your RNA binding protein, as is the case here. It can be common practice to characterize it in phosphate buffer to stabilize it. But then what one then observes, one can see that there's millisecond motion, or excuse me, there's REX. I should distinguish these two. There's REX present that represents the phosphate buffer binding on and off at a millisecond process, but it's not intrinsic millisecond motions. And so Dyno1 didn't know anything about that. So it's really interesting to me now that we seem to have enough sufficient predictive power for some of these properties that we can kind of even use it in a way to query the data itself of like, hey, is this real motion? Could this be some sort of artifact? And furthermore, we are also interested, so in the big guns of NMR CPMG experiments, which, if you're familiar, can be a more precise way to query millisecond processes over the other types of relaxation. Dyno1 had predictive power and it predicted millisecond motion in some residues that by conventional means of analyzing CPMG, the conventional data processing would have missed. And that essentially, and I'm happy to talk more about this offline, is where there's unsuppressed R2. And if we then we revisited how we did our data processing pipeline and considered that unsuppressed R2, that our predictions actually then went up. So it's really exciting. We're in a position now. So in my postdoctoral work, through curating these different experimental benchmarks of millisecond motions at different quality contents and different numbers of proteins, we were able to then train a model that we named Dyno1 on this missing BMRB, and we see that it has predictive power for proteins with more careful, more high quality experiments done on those millisecond motions. And our rationale for why language models can do this is that we saw that these different timescales have different evolutionary patterns underlying them. And from my point of view, it makes a lot of sense. Something we know about language models in general is that they are good at picking up on motifs. And so in some ways, these collections of residues with concerted motions representing millisecond motions, those also are motifs. And so very excited about future directions for all sorts of future extensions for Dyno1. And so people to acknowledge for helping this project. So Gina was a joint first author on this. Ramith, Sergey, Lucy Caldwell and Dora have all been really instrumental in making this possible. And also need to thank anyone who's ever done an NMR experiment and deposited the data, because that is all now part of these data sets, in addition to folks who have been very kindly worked very hard to dig out some of these old relaxation data sets. And I'll stop there, and I'm very glad to answer questions.
Thank you, Hannah. This is beautiful. I'm always fascinated, and I think it's one of the key questions these days. It's like, how do we start integrating more and more dynamics in our design pipelines and in our prediction pipelines? So in that direction, Amit asked if you ever considered including beta factor data, and so he comments that while you may need to normalize for resolution and perhaps ignore TLS refined XRD, there might be some use in looking at structures with higher B factors to see how that correlates with millisecond data or RX data.
Yeah, so there was a slide in there that I might have glossed over, but so what we saw was that there was basically no correlation between where there are high B factors and where there's missing peaks. And so that is really interesting then, because it suggests that they're totally like orthogonal types of data that could be very useful to use together. And a next direction that we're very excited about is combining NMR, training from NMR data with other data types as well. And those sorts of questions will be at the core of how we do that.
Great. Bruno, you sound like you have thoughts and questions.
Totally. Let's see. So I guess, Anna, maybe one question for you, and this is more so like just a general, like, why are millisecond motions more relevant to function than other time ranges? You know, I guess, you know, in your mind, this seems very clear. This sort of like, I mean, like this millisecond thing is what matters.
Yeah, so let's talk about thermal fluctuations, which happen, right? And so there's some background of thermal fluctuations that can just be there, will just be there.
Yeah, that's right. It will be there, right, you know,
yeah.
by definition.
And so the millisecond processes that we view as being would be really interesting to be able to predict often involve basically multiplying probabilities of multiple things happening together. So it's milliseconds, not because it's actually like slow, like it takes milliseconds, but it has a rate of milliseconds, so that it's sort of waiting for a number of different random fluctuations to happen at the same time. And so then it's like boom, 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 they can all happen together. And then that causes this shift. And so those types of collective processes are often related to function, because it's really how do you encode going from, you know, that collective action to go from one state to something that is significantly different, versus thermal things might sort of gradually take you from one to the other. And that could happen, those could happen on a variety of timescales.
I see.
Yeah, Liz, I see you.
Because you did say, so it seems to me logical that you can maybe detect those slower ones more easily because of the nature of those motions, but you had correlations also with the faster ones, like they're there. It's just a bit weaker. So I suppose it's about how do you bridge the timescales, right? How to go about, because, and this is also, you know, stuff from Dorothy's lab before and others. How do we, when will different timescale motions be related to each other, and when will they not? Right?
Yes.
That's sort of
yeah.
the question.
Yeah. And I think also the thing that interests me is, you know, because some of these are and you know, there's definitely going to be evolutionary pressure to cause the functionally relevant ones to be there, but then there's also times where people, myself included, like we see a process in NMR, it happens to be there, we didn't actually know it would be there, and it also happens at a millisecond process, but we don't know if it has functional relevance. So in specifically for in the Chi-B fold switcher, it actually has a fourth state that we have no idea what it's doing there, and we observe it in vitro. We don't know if it exists in vivo, but there's also that question. So there are also, like, it's I would hate to be saying there's a strict like one-to-one between millisecond processes and biological relevance, and so then it's how do we keep teasing out those questions.
And maybe amide exchange too, right? As another orthogonal one. Obviously, there's so many. I think there's
Yeah.
to add in, but there's quite a lot of data there already. So.
Yeah, and so yeah, going forward, I'm very excited to see how we can, or for instance, like stability measurements in high throughput, like from Gabe Rockland's lab. How could types of these be pulled together? As well for predicting protein-protein binding. That was a question I had for Minkyung. I'm really curious. I haven't worked on protein-protein binding at all, but could one imagine just taking a bunch of KDs and structure data and like using those in combination? Like that sounds hard, and I'm sure people have thought about doing that, but like what would be the barriers there?
I mean, so for the protein-protein interactions, I think that there's a lot of bars, especially for the interactions for some flexible regions, I would say. So it's really hard because the current protein-protein interaction prediction models, especially for the direct interaction prediction model, they usually relies on the structural data bank, the PDB. But in PDB, most of the structures are actually focused on ordered proteins. And for the IDR protein interaction kind of thing, they usually just use the fragment, the peptides of the IDRs to determine the 3D structure. So actually we don't know how the entire protein, the full length protein interact with each other just based on the PDB. I think that's the one thing. And yeah. So the other thing is, I mean, it's somewhat a kind of question to you, Hannah, but in the protein ensemble structure prediction or protein flexibility prediction methods, I'm not sure whether they are focusing on functional state or just some thermodynamic ensemble structure prediction. So to answer the most of the biological question, I think it's important to predict the functional state rather than just thermal fluctuation. And I'm really curious whether the NMR signals or other approaches can distinguish what is just thermal fluctuation and what would be more functional relevant movement.
Yeah, so I was attracted to NMR because I didn't see a better method for characterizing a complete landscape of all of the motions. That said, even if you see the complete landscape, that itself doesn't tell you what is the biologically relevant motions without doing further interrogating of your system.
Yeah, I think there are some related questions also in the chat in terms of like, how does the peak assignment then depends on the presence of ligands and substrates or the assignment of absent peaks. And how does it then also change depending on the size of the protein? How much does that matter and how your data set got curated for that kind of stuff?
So for the first question about ligands being there or not, for Dyna1 curation, I took the approach of I'm just going to take everything in the BMRB. And so some of those are in complexes, some of those have ligands bound or free. But I sort of wanted to just not think too carefully about that part. We, for our validation set, had things we had carefully curated to be only in their apo form, but we just sort of threw in everything. And so there's many cases where somebody might have a binding site with millisecond motion. The binding site is exchange broadened. When you titrate in a ligand, it then stabilizes the interaction and the peaks appear. So in principle, Dyna1 was trained on all that. And I think like how AlphaFold was trained on sort of everything in the PDB, we're now seeing that if we try to disentangle different attributes, we can get more utility. And so that's sort of a nature of things we want to explore and keep exploring. About the size limitation. So yes, there's more things that are size 100 than size 300 in the training set. But I want to find more examples of big proteins for which we know information about millisecond motions, because my sort of knee-jerk reaction is, oh, we should probably have a, your mileage may vary sticker on our model for bigger proteins. But then also I tried running Dyna1 on a massive protein that somebody contact studies, and it does distinguish things. Like it's messier than other things, but it does still have predictive. So I would love to work with people who do think on bigger complexes and to try to figure out like what is known about millisecond motions in those bigger ones. What would we expect? Is the model failing? What would we like the model to do better?
I'm gonna.
I think this also. Sorry,
one
Liz.
more in there. Sorry, I know I'm interrupting, but I feel like something for all of your talks that I was wondering about was you can look at the whole system, but what about just a mutation, one mutation, where you really have the data, right? Can that, I mean, people are trying to do better with those, of course, as well. But when that's sort of complementary, you can try the whole thing and then dig deeper. And anyway, it's a comment.
Well, maybe I can talk to that one. I guess there has been sort of like this, like, incredible promise in the field that we will just go and site saturate proteins and learn things about them. And I think this exists, and I think it's real. And people do collect these data sets. It just turns out that we have not seemed to be able to translate that into improved models, I guess, for design or for prediction, for all that matter. And I think part of it is because oftentimes, well, I don't know if it's the full answer, but part of it is because oftentimes when things start failing or succeeding, you don't really understand why, and they can fail and succeed. So you can get like, so let's say that you make a point mutant and you just say like, okay, well, and this is in a protein-protein interaction, right? And then your output is just saying, okay, the protein interaction was disrupted. Okay, but was it disrupted because it was disrupted because of binding or because the protein no longer folds? And I think this is sort of like, I think it's just kind of hard to disentangle exactly. And I'm not saying that there aren't systems and ways to do it, but, every time that you want to know deeper and deeper why it is, it becomes a little harder. But I think it's still a challenge for us, right? Because I think, like somebody says, these days kind of like collecting data is no longer a problem. I think the problem is to make sense out of it. And or at least in some of the things we do is no longer a problem. In some other, like NMR, maybe it's still difficult. But if you see what I mean, right? And I think this is like it's almost like a collective failure of the field, or it's not a failure, it's a challenge for the future. Like, so like what can you do with these data points, right?
Liz, we've been very interested in the question of mutations, but something that I'm finding a challenge is that there's sort of well-known examples of point mutants and that perturb dynamics, but I haven't yet found an example of a point mutation that totally like creates or destroys a process.
But I think they exist. I think they exist.
I mean, I'm looking, and I would love if there are any pointers to because often like mutations can change the rates or they can change the populations of a process, but they don't necessarily create or destroy a process.
Probably Liz has a few examples, right? In sort of like in her studies.
Yeah, so if there are things they can point me to, that would be fantastic, because we're very much trying to find things like that that we would know if you make this thing, it should like kill it, versus just if you make that, it should change the rate or population.
I think I'm like in our systems, I think we observe what Hannah is saying, like in a lot of the cases, like doing one mutation, it's just like shifting things a little bit. If you even add several mutations, we don't see like a complete kill of the dynamics. It's more of a shift towards a little bit more rigid or a little bit more flexible, but it has been a bit also like a struggle to find really that perfect sauce. But I guess it's always system dependent, right? Which kind of brings to the question of like, I think the dream is really that we start designing dynamics and more towards like systems that can use these dynamics in a functional manner. And so I know Bruno has been doing some work in that direction. And I think this is something to reflect on. How can we get there? I know recently, Bruno, you came with this idea that, like, you know, what we should be designing for, it's not the 99%, it's the 1% that actually gives you the functionality
Yeah, I'm not
that
sure
you
I
need.
can put data in my words yet, but at least but yeah, I think, you know, sort of like, I think that idea is basically what characterizes how biological systems work, right? You know, not every biological system, but the fact that they can access some alternative state, and then that they have a trigger that basically leads them to that alternative state, right? You know, in a simple embodiment of thinking about this is sort of like, I guess, when you have a peptide in its unbound form, does it sample the bound form, right? So like, so there is, it already has some kind of propensity to sample that bound form, but then when it finds its binder, it locks, right? And I think nature does this like over and over and over again, and it encodes these kinds of things in sequences. I think, you know, in terms of design, we're complete clowns doing these kinds of things. Of course, there has been like a few people that have done it, you know, successfully, particularly Tanya, David, you know, and there's a few, you know, Roberto also has done a few things. Um, but the things have been very far in between. And I think this idea that you can actually sort of like say, oh, like, you know, let me just sample 2% of this state. I mean, that's a dream, right? To me, at least at this point, or 1% or whatever. Now, one of the questions that I like to think of is like, you know, how much is important to populate a particular state to sustain a biological function? I think this to me is interesting, and I think here design perhaps might help us to get to some of these questions. You know, and actually, I think, well, one thing, I think when you have a population of 2% or 10%, let's say, I think point mutants actually can make a big difference. It depends to the length, you know, it depends on the lenses that you're using it. And then, of course, you know, the stuff like Anna is doing, like NMR, right? You know, it has this like very good lenses to see if the population of 10% that is so important for function, if you mutate one thing, reduce it to like 0%. I don't think it's unthinkable.
Well, Dino
Yeah,
wants
it's
to
interesting
find,
sorry.
if anyone wants
Go
to
ahead.
put it in their design algorithm, I'd love to hear about it. Love to hear if anyone has success like designing it into a backbone or something. Sorry, Alena, I cut you off.
No, I was actually just gonna add, like, it's interesting you're talking about like how you populate states, because that's what we've been seeing in our assemblies, right? Like, so we've been trying to get away from symmetry, and then you need to have some flexibility in the building blocks to access different type of architectures. And there it seems that it's really like dependent on how much you can sample particular states. Unfortunately, there is not a lot of very good methods for us because our building blocks are actually pretty big. So Hannah, maybe we can challenge you and send you over some building blocks and see what you can get out of it. Because we would be curious to see experimentally if, like, we typically sample these with MD, right? So molecular dynamic simulations are still like our tool to go. And we'd love to get to complement it with something that would be really experimental, that because seeing is believing, right?
Mm-hmm. Yeah, I'd love to chat more. It'd be interesting to hear.
So I think it's amazing, actually. People are really staying, and so we've gone actually half an hour longer than we had planned, and people are staying. I think there is such incredible scope in the work that you have been presenting, and I think maybe I'll draw it to a close. I don't want to, Ilona, I think this is always the problem, is we would like to actually, this online format doesn't let us chat indefinitely, even though we might love to. I might put it back to you. I mean, I feel like we have, is there anything anyone wants to still add at the end before maybe we close? I'll turn it to you, Ilona.
I don't know. You guys tell me. I'm sure that everyone has thoughts.
I know.
It's
I think I
always
have
the
a
problem
challenge.
when we get together.
Yeah, I think I have a challenge for Minkyung. You know, like, so, you know, could we actually just do like an all against all proteome sort of like, you know, pairing? Or when could we actually do that? Because, you know, in the end, protein interactions, I mean, they depend on a lot of things, local concentration. So if you actually push like real high systems, like how many things are there that are not really encoded in evolution that we might still be able to discover, and with which confidence? I mean, because getting docked complexes is one thing. The fact that they exist is a whole other thing. And then of course Anna can validate them in the NMR2 is going to be fine, so it's no problem.
High throughput NMR is just a couple years away.
Exactly. Yeah, months in fact.
Yeah.
Yeah.
Yeah, sounds good.
All right.
Let's start.
I think we
Perfect
have
plan.
much to do, and I look forward to the next webinar. I want to thank you all too. You've done an amazing job answering in the chat, and thank everyone in the audience for their questions as well. I think this set of topics that we had here is just such an exciting frontier, and I thank you all again for being here. I'm excited to see where we end up next. Thank you.
Thank you, Liz, for hosting us.
My pleasure.
But
Okay, take care, everyone.
bye.
Bye.
Bye.